Okay, hello. See me very much. Okay, let me finish chapter 2. Okay. So last time we finished talking about the different type of molecules, or different type of biomolecules. Okay. Biomolecules by themselves, there are units of biomolecules by themselves, but there are other combinations of them. For example, you could have a glycosylated protein, which means there's a protein with sugars attached to it, and that's really common on the cell surface, like blood typing molecules and stuff like that. Lipoproteins, okay, proteins with lipid components attached to it, like LDLs and HDLs. We talk about cholesterol and stuff like that. Okay, all right. Okay, functional groups. Okay, these are different types of functional groups. Okay, functional groups are small units. Okay, of elements that move together usually as a unit, like something like a hydroxyl group or a amine group. Like these groups are usually work together, and when you cut them as a unit, and when you add them as a unit. Okay, like I'm sure you heard of ATP. ATP turns into ADP because you lose a phosphate group. Okay, so these 4 functional groups are the most common. There are actually many more than these, but these 4 you do have to know. Okay. 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 Electrons. Okay. So about what electrons do. Okay. Okay. So electrons have 4 important biological roles. They form covalent bonds, they form ions, they form high energy electrons. So they're used to transfer energy from one place to another. And then last, they form free radicals. And that's actually a byproduct. That's actually damaging for the most part. Okay. Okay, so atoms are by definition neutral. You can take an atom and you can add electrons to it, or you can subtract electrons from it. Okay, when you add electrons to it, okay, when you add electrons to it, they become negatively charged and they become an anion. Okay, so these are the common anions that you have to know. And then if you take an electron and remove an electron, you get a cation. And these are the cations that you have to know. Okay. Atoms and molecules. Okay. Okay, so this is an atom. Okay. Atoms, okay, have 3 parts to them: protons, neutrons, electrons. The protons and neutrons are inside, okay, inside the nucleus, and the electrons are outside the nucleus. Okay, the number of protons determine the element. Okay, so you add electrons, or sorry, if you add protons, okay, they change the element itself. You change the fundamental nature of the thing that you're working on. Okay. If you change the neutrons, okay, you change the isotope. Okay, so for example, carbon with 6 neutrons. There are carbons with 8 neutrons. There are carbons with 7 neutrons. Okay, all of them are still carbon, but what you change is the isotope. Okay, they're slightly different weights. Okay, and some of the isotopes are radioactive, which means they can break down and stuff. Okay, all right. And electrons will cover in more detail. Okay, so this is an isotope, right? So this right here, okay, is your typical hydrogen atom. Okay, it has no neutrons. Okay, if you add a neutron, it's still hydrogen. It stays hydrogen. Okay, but now it has one additional neutron. Okay, so it is a hydrogen isotope. Okay, it's deuterium. It's called deuterium, but it's still hydrogen. It works just like hydrogen. It does everything hydrogen does. It just weighs more than hydrogen. Okay, if you take this and you lose an electron, so there's no more electron here. Okay, all you get is H plus. This is an ion. Okay. All right. So these are the common biomolecules with that. Okay. I'm not sure why they put the material on the front of the chapter. Okay. Okay. Covalent bonding between atoms create molecules. Okay, a molecule is something that has covalent bonds. Okay, these covalent bonds means that these things are sharing electrons. Okay, if they're sharing electrons, okay, they share at least one pair of electrons. It takes one pair of electrons to make a single bond. It takes two pairs of electrons to make a double bond. It takes three pairs of electrons to make a triple bond. Okay, and these bonds hold the two atoms together tightly, and so they move together as a unit. And it requires a lot of energy to actually make them, but once they're stable, it requires energy to break them as well. Okay. Okay. Um, there are two general types of covalent bonds. What they're nonpolar and polar. Okay. So imagine a tug of war where two sides are about equal size. Okay. If they're about equal size, they'll pull the electrons toward them equally. And therefore, one side won't be able to win over the other. Okay. And that is a non-polar bond. Molecules with only non-polar bonds are non-polar molecules. Okay, a polar molecule has at least one polar bond. Okay, so water is like literally the best example of a polar molecule. Okay. Okay. Hold on one second. Okay. Okay. Sorry, I had to take a little break. So polar molecules. Water is probably the best example of a polar molecule. Okay. The oxygen is considerably stronger than the hydrogen when it comes to electrons. Okay, so it wants the electrons more. So it's going to pull the electrons toward it. Okay, so that means the hydrogens are going to not have that many electrons. Okay, electrons are negatively charged, so there's going to be a negative pull this way. There's going to be more electrons on this side and more positive charges on that side. Okay, so this is plus, okay, and then this side is minus right here. Okay, and so they're not going to be equal to each other. Okay, and so these will be polar molecules. Polar means the two sides are not equal to each other in terms of charge. Okay. Okay. Ions are charged atoms. We've already talked about that. Okay. Okay. Non-covalent bonds, okay, are much weaker. They're about an order of magnitude weaker than covalent bonds, which means they're about 10 times 1/10 the strength, which means they're easier to make and they're easier to break. Okay. Ionic bonds are electrostatic interaction between ions. Okay, atoms either gain or lose electrons, and then the anions and the cations attract each other. Okay, opposite charges attract. Okay, hydrogen bonds. Let me see. Let me show you a picture of hydrogen bonds. So this is a hydrogen bond. Okay, hydrogen bonds are going to be the positive hydrogens, okay, on water. Remember I said the hydrogens are slightly positive and the oxygens are slightly negative. So the oxygens are slightly negative, which they'll attract the positive hydrogens on the other water molecules, and the hydrogens will attract the oxygen molecules on the other water molecules. So they pull them in together, okay, and that bond a hydrogen bond. Each individual hydrogen bond is very, very weak, but hydrogen bonds are really important in biology, okay, because there's so many of them. They make up for its weakness in terms of sheer numbers of hydrogen bonds, okay? Almost every single molecule I think of in biology has hydrogen bonds and other stuff to form structures. Okay. Okay. Hydrophilic interactions create biological solutions. Okay, so our body, okay, is about 70% water, right? I'm sure you guys know that from like PBS specials and stuff like that. Okay, that's true. Our bodies are about 70% water. Okay, and therefore, solutions in our body deal with water and things that are dissolved in water. Okay, a solution is something that's so a solution has two parts, a solute and a solvent. A solute is usually something that's dissolved. So a solute is usually solid, but it's dissolved in a solvent. Okay. So biological solutions are all water-based. So ions, like salt, okay, calcium, etc., are dissolved in water. Okay, That leads to a concept called solubility. Solubility is the ability of a solvent to dissolve in a solvent. Okay. Depending on the solvent, they'll have different solubilities. Okay. Like for example, you can take salt, you can dissolve it in water very easily. Okay, so salt has a very high solubility in water. You take that same salt and you try to dissolve it in oil, and it won't dissolve nearly as well. So salt has very low solubility in oil. Okay, things that are let me rephrase. Things that are solutes that are hydrophilic are going to be very soluble in water. Things that are hydrophobic, okay, non-polar molecules will not be soluble in water, but it will be soluble in things like that are also non-polar, like oils. Okay. Okay. So there we go. Solute, solvent, solution. Okay. The next concept is concentration. Okay. Concentration is the amount of salt or the amount of solute in the volume of solution. Again, and this actually you guys will go over in more detail in lab. Okay, so if you guys haven't already. Okay. All right. Okay. So the mass of the solid before it dissolves is usually given grams or milligrams. Okay. In physiology, like in all science, we use grams and milligrams. We do not use pounds and stuff typically. Okay. The molecular mass, okay, is derived from the chemical formula of the molecule. Okay. So molecular mass of something like my pen. Okay. The molecular mass of H2O, for example, a really simple molecule. So oxygen, the mass of oxygen is 16. Okay. The mass of hydrogen is 1. So there's 2 of them. So 1 plus 1. So the molecular mass of hydrogen of water is 18. Okay. 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 So more concepts that you can read out this slide. Make sure you read this slide actually, but I mean, I don't want to just spend this time reading this out for you guys. Okay. 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 Expressions of volume. Again, you guys will be going over this in lab. So we'll skip this figure. Okay. Percent solutions. Again, we'll be going over this in lab. Okay. Hydrophobic interactions. Okay. Hydrophobic interactions. So the way we dissolve, the simple term to explain solubility is like dissolves like. Polar molecules will dissolve in polar solutions like water. Okay. Non-polar solutions will dissolve non-polar things like oils and fats. Okay. All right. Let me go to shape. Okay. When we talk about this is true when we talk about anything, but especially true when we talk about protein. Okay, all proteins have a shape that's controlled by a whole bunch of different interactions with temperature, with water, the solvent that is in, with the pH that's in, etc., etc. And that leads to a shape. Okay, it's not a big blob of something. It actually has a shape. That shape is incredibly important in determining what function it has. Any alteration in shape, and the protein can lose its ability to do whatever it's supposed to do. Okay. Okay. So we already talked about this: the alpha helixes, beta sheets, tertiary structure, quaternary structure, etc. Okay. So we were talking about this in earlier recording. So make sure you go over what a primary structure, primary, secondary, tertiary, and quaternary structure is. Okay. All right. Next concept I want to make sure you guys understand is pH. Okay. pH stands for the potential of hydrogen. Okay. So pH is a measurement of the number of free H pluses in the solution. Okay, pH is literally the negative log of the number of hydrogens, or the concentration of hydrogens that are in the solution. Okay, a pH of less than 7 is acidic. By acidic, it means there's a lot of hydrogens in the solution. A pH of greater than 7 is alkaline. Alkaline means there's very few hydrogens in the solution. Okay. Okay. Our blood has a pH of 7.4. Okay, which is slightly alkaline, and it has to stay that way. And it has to stay that way, or else we die. Okay, so our blood has buffers. Okay, that buffer moderates the changes in pH. It keeps the pH at the exact level, in the middle. So it doesn't go too high, it doesn't go too low. Okay. If you think about this way, if we didn't have this bicarbonate buffer in our bloodstream, drinking a Coke would actually kill you. It would drop the pH in the blood way too low, and it will kill you. Okay, but because we have buffers, these buffers can absorb excess hydrogens when we need them to, and can release these hydrogens when we need them to. So it keeps the pH at a very steady level. Okay, that's what a buffer does. Okay, so acids and bases. Okay, if you're really into chemistry, there's many different definitions of what acid is and many different definitions of what base is. In physiology, things are much simpler. Okay, an acid is something that contributes an H plus. A base is something that can absorb an H plus. Okay. Okay, so an acid is something that gives off the H plus. A base is something that absorbs the H plus. Okay, so something that absorbs the H plus. Okay. Okay, so definition of what a pH is, right? It should be potential hydrogen, not power, but So pH is equal to the negative log of the H plus concentration, or written another way, pH is equal to the log 1 over the concentration of H plus. These two are equal expressions. Okay, so but in other words, when you look at this actually makes a lot more sense. When you have 10 times the amount of hydrogen, you decrease your pH by 1. Okay, so something with a pH of 2 has 10 times the hydrogen ions as something that has a pH of 3, which has 10 times the hydrogen ions as something that has a pH of 4. Okay. All right. So 10 to the 7th H plus is literally pH of 7. You calculate all this out, it's a pH of 7. Okay. I will not ask you guys to calculate pH values, okay? But you do need to know what's acidic and what's basic, etc., etc. Okay. All right. Protein interactions. Okay, so proteins. I kind of glossed it over at last chapter what they do, but proteins do pretty much everything inside the cell. All the chemical reactions that occur inside the cell, most of it is actually done by proteins. Okay, number one, enzymes. Okay, membrane transporters, signal molecules, receptors, binding proteins, immunoglobulins, regulatory proteins, and there's more than that. Okay. Okay, so safe to say, proteins interact with things inside the cell, and therefore there are going to be binding sites where things attach. Okay, all right. So let's talk about what binding sites are. Okay, so this is a protein. A protein will have a site that binds to things, and the things that bind to it will be called ligands. Okay. Okay. The best model is actually this model here. It's called the induced fit model. Okay. The induced fit model is the best way to explain what an induced fit model is shaking hands. Okay. As you shake hands with someone, you extend your hand out, right? You extend your hand out. But when you extend your hand out, as you grab someone's hand, your hand has to change shape. So you can grab the other person's hand. The other person's hand also has to change shape, so they can grab your own. So it has to induce fit. The fit is induced by the head. Okay. All right. Okay. So some terms. Okay. Affinity is the degree to which a protein is attached to the ligand. You think of this as how much a protein wants to hold on to something. Okay. Equilibrium is binding. The rate of binding is equal exactly to the rate of unbinding. Okay. That's when the protein spends about equal time binding and like bound and unbound states. Okay, and that is determined by a constant called equilibrium constant. Okay. Okay. There's a typo here. Okay. It should be KEQ. That's not a G. That should be EQ. Okay. CK, EQ. Okay. And then there's a typo here. Okay. Okay. So here is the law of mass action. The law of mass action says when protein binding is in equilibrium, the ratio stays constant. Okay. Not the units. Okay. The ratio between the forward reaction and the backward reaction is the same, and therefore it stays constant. Okay. So the rate of things going forward and the rate of things going backwards stays constant. Okay. When we add this, when we add more of P, okay, then we can drive that reaction forward. Okay. Until it becomes constant again. Okay. So if we add more on this side, then that reaction will, this forward reaction will happen faster. The backward reaction will happen slower. So eventually we'll have more of the, okay, so let me put it in terms of colors. When we add more blue stuff, the forward reaction will convert it to more orange stuff until they're equal again. Okay. And then everything is restored. That's the law of mass action. How we actually use it is actually in reverse. Okay. In biological systems, a lot of times we drive the reaction forward. We want things to move quickly. So we drive the reaction forward by using up all the orange. You use up all the orange, the blue will be driven to make more orange, and we can use up all the orange, and the blue will make more orange. So we keep chemical reactions going faster by the law of mass action. Okay, just driving this reaction forward. Okay. In chemistry, this is if you guys have taken chemistry, this is known as Le Chatelier's principle. Okay. Okay. So the dissociation constant, okay, tells you how likely something is to break apart. Okay, so it's actually literally the opposite, the inverse of KEQ. Okay, if you see this, the dissociation constant is literally 1 over these two flips. Okay. Okay. More definition: competitors and agonists. Competitors are related ligands that compete for the binding site. Agonists are competing ligands that mimic each other's actions. Okay. So oof. Okay. Multiple factors affect protein binding. Isoforms. Okay. Isoforms. We'll talk more about this when we talk about DNA, but isoforms are closely related proteins that are usually made from the same sequence of DNA. Their function is really similar, but the different affinity for the ligands are different from one another. So some are stronger and some are weaker, and depending on how many of each one you have, that determines how much binding you have for the ligand. Okay, a lot of this next one is probably the most important one. Activation. Activation. A lot of proteins are sitting around in your cells and they're not doing anything because they're not activated. Okay, and they don't get turned on until they're activated by either proteolytic activation or cofactors. Like proteolytic activation is usually cutting protein lytic cutting. So you cut off a little bit of it to activate it. Okay. The best way to think about how proteolytic activation works, it is how grenades work. When you have a grenade, you have a firing pin. As soon as the grenade is pretty much inert until you remove the firing pin. You pull out that firing pin, so you cut out that little piece of protein, and then the protein works. Okay, and then the grenade works. Right. So, okay. Okay. So the chemical modulators, competitive inhibitors versus non-competitive inhibitors or reversible antagonists. Okay. So 5, 2. Leaves a little bit more. Okay. So this is proteolytic activation. You take an inactive protein, you cut off those sequences, and now all of a sudden this becomes active. This is insulin, by the way. Okay. So this is how insulin works. You make it, it's inactive. All you have to do is remove these two little pieces, okay, and it becomes active. Okay. Okay. Some proteins require cofactors to work. Okay, so without the cofactor will not work. That's where a lot of vitamins and minerals actually come into play. A lot of vitamins and minerals that you eat are actually cofactors for different enzymes in your body. Okay, allosteric activators. So this is an active protein. So we can make all of this we want. Okay, you can have all the ligand you want, but the ligand will not bind to the inactive protein until you make an allosteric activator. Okay, and the allosteric activator binds to a different site than the ligand, but once it binds, it changes the shape of the binding site. Now the binding site is now like perfect and can fit in the ligand. So I can make as much of this as you want, okay, and you do, and all you need to do to turn everything on is to make the little tiny piece, the allosteric activator, and then it turns on, okay? So also, if I destroy this piece, then it turns off, okay? Okay. Okay. So there's a lot of different drugs that do this. Okay, a lot of drugs are competitive or allosteric inhibitors of certain things. A competitive inhibitor will bind to an active site of a protein, of an enzyme. Okay, and it will prevent the enzyme from working. Okay, it's competitive inhibitor because it competes for the same site. Okay, it binds to the active site, so it competes for the same site as the ligand. Okay, a non-competitive inhibitor or allosteric inhibitor binds at a different site than the ligand does, but once it binds, changes the conformational shape of the protein, so it can no longer bind to the ligand. So you can't compete this away. Okay, competitive inhibition can be overwhelmed. Like competitive inhibitors, all I do is add a lot more ligand, and I can outcompete competitor. But in this case, it doesn't matter how much ligand I have. If this is here, I can't do anything at all. Okay. Okay. Other factors that activate protein binding: temperature and pH. Okay, that is very, very, very important. Okay, you change the temperature, you change the pH. Okay, the protein will fold differently, and when it folds differently, the binding site may or may not be there, and therefore it may or may not work. Okay, you change the temperature or pH dramatically from where it's supposed to be, then for sure it doesn't work. Okay. All right. Enzymes are not magic. They make things occur millions times faster, but there's a limit. The site has to have ligand in it. So if you add more ligand, that site can work faster and faster and faster until you reach a limit where it's working literally as fast as it can. When it's working as fast as it can, that enzyme is saturated. Okay, that also means we have reached the maximum reaction rate. That enzyme, every enzyme in that solution is working as fast as it can. If you want to increase the reaction rate from then on, you have to add more enzyme. Okay. So here, so if I have an enzyme in solution, if I add more ligand, it works faster and faster and faster until I reach saturation. Now, no matter how much substrate I add, or ligand I add, I don't get a faster reaction. If I give you one thing to do per hour, you can do it. If I give you two things to do per hour, you can do it. If I give you 100 things per hour, you can't get everything done, even though you get as fast as you can. Okay. So this is changes in temperature and pH. Like I said, small changes in temperature will affect the activity a little bit, but not dramatically. But dramatic increases or decreases in temperature will cause it to not work at all. Okay. All right. Hope that all makes sense. Okay. I'll see you guys for chapter 3.